All living organisms require a continuous supply of energy for their survival. Energy is carried out various functions such as uptake of nutrients, absorption, growth, development, movement, reproduction, and even breathing. About 50% of energy produced by cell is utilized by these cellular activities and rest of it's changed into heat and is lost into environment. Now the question arises from where does this energy to carry out all these processes of life happens. In this chapter we are going to deal with the basic concepts of energy producing biological process called cellular respiration. In the cell, how a respiration takes place. Majority of people I know, they don't use their cells at all. They just keep idle, watch the movie on the TV or play games. We eat food in order to obtain energy. The food we eat is in the form of macromolecules and is oxidized to fulfill energy requirement of our body for carrying out all the basic life processes. As we have already studied in the previous chapters that only green plants and some bacteria can prepare their own food by the process called photosynthesis. They obtain their food by converting light energy into chemical energy, which thereby gets stored into the bonds of carbohydrate molecule such as glucose, sucrose, starch, etc. But all cells, tissues and organs in plants do not photosynthesis. Instead, photosynthesis takes place in only some parts of the plants. I repeat, in only some parts of the plants. That is, only cells that contain chloroplast, that is the green parts of the plants. Hence, all other non-green parts of plant need food for oxidation to get energy. Thus, food has to be translocated from the green parts to non-green parts for oxidation process to generate energy. Okay. Now let's move on. Animals, on the other hand, are heterotrophic in nature. They either obtain their food directly from the plants, that is herbivores, or indirectly depend on the herbivores for their food, that is carnivores. Saprophytes are dependent on dead and decaying matter for their food. Example, fungi, some bacteria. Thus, it can be concluded that all food that is required for life processes ultimately comes from photosynthesis. So, photosynthesis is the biggest life process of biological world. In this world, photosynthesis is the biggest thing that happens. And it's just a wonderful thing. Cellular respiration is the process of breaking of C-C bonds of complex compounds through oxidation within the cells, leading to release of considerable amount of energy in the form of ATP. Considerable amount of energy in the form of ATP. It is also noted that site of breaking down of complex molecules to yield energy in the cytoplasm and mitochondria of the living cells. Plant cells also carry out cellular respiration to get energy. In the next slide, what we are seeing is the compounds that are oxidized during the process of respiration are called respiratory substrates. They are called as respiratory substrates. Carbohydrates are used as major respiratory substrate by most of the organism to get energy. But under some condition in some organism, protein, fat and organic acid are also used as respiratory substrate. During the process of oxidation of food within a cell, all the energy contained in the respiratory substrate, right? All the energy contained in the respiratory substrate is not released. Energy contained in the substrate is not released free into the cell or in a single step. Instead, it is released in a series of stepwise reaction controlled by enzymes and is trapped as chemical energy in what form? In the form of ATP. Thus, it is said that ATP acts as the energy currency of the cell. The energy trapped in ATP is used in many energy requiring processes of the organism, and the carbon skeleton produced during respiration is used as precursors for the biosynthesis of other molecules in the cells. The energy trapped in ATP is used in many energy requiring processes of the organism, and the carbon skeleton produced during respiration is used as precursors for the biosynthesis of other molecules in the cells. Precursors for biosynthesis of other molecules in a cell. Now, let us look at the process of respiration takes place. Oxygen and release carbon dioxide. Plants have stomata and lenticles for gaseous exchange instead of specialized organs that are present in animals for exchange of gases. So what are the reasons which show how plants can get along without respiratory organ? Plants do not have lungs. How can get along without the respiratory organ? Every part of the plant has the ability to take care of its own needs of gas exchange, and also very little transport of gases occurs from one part of the plant to another. Next, when cell photosynthesis, availability of oxygen is not a problem in these cells due to continuous release of oxygen that takes place within the cell. Also, in cases of stems which are thick and woody in nature, the organization of the living cell is in the form of thin layers which are found inside and beneath the bark, like leaves which have stomata for gaseous exchange, right? These stems also have openings called lenticles. Internal cells are dead and provide only mechanical support to the plant or tree. This depicts that most cells of the plant have at least a part of their surface in contact with the air. The loose packing of parenchymal cells in leaves, stems and roots and provides an interconnected network of air spaces which help in facilitating this process. Now let's look at the cellular respiration may be classified into two types. Okay, one is aerobic respiration and another one is anaerobic respiration. Aerobic respiration when there is oxygen. Anaerobic respiration in the absence of oxygen. There are some exercises, we do aerobic exercises. When there is no oxygen involved, it is called as anaerobic exercise. Similarly, aerobic respiration is the type of respiration in which organism utilizes oxygen for the complete oxidation of organic food into carbon dioxide, water and energy. It starts in cytoplasm and gets completed inside the mitochondria. Anaerobic respiration. In this type of respiration, which organic food is oxidized incompletely, it occurs in cytoplasm and releases small amount of energy. It is believed that first cells on this planet lived in an reducing environment when oxygen was not present in the atmosphere. Listen, oxygen was not present in the atmosphere. They were anaerobes. Some of them are facultative anaerobes while others are obligate anaerobes. Thus, in all cases, all living organisms retain the enzyme machinery. They may retain the enzyme machinery for partial oxidation of glucose in the absence of oxygen. Partial oxidation of glucose in the absence of oxygen. And this breakdown of glucose to pyruvic acid is called glycolysis. Lysis breakdown, right? It occurs in cytoplasm of the living cells. Let's move on. Cellular respiration occurs in all living cells and proceeds with the help of the enzymes. Okay? So that's the fundamental. The first step of respiration is the glycolysis, after which is pyruvic acid may enter the Krebs cycle, which is aerobic respiration, where we need oxygen. Or they undergo fermentation, which is anaerobic respiration, where they do not need oxygen. Let's see glycolysis. The scheme of glycolysis was first introduced by Gustav Embeden. It is referred as EMP pathway. The EMP comes from the people who found this pathway. It is the common pathway in both aerobic and anaerobic modes of respiration. But in cases of anaerobic organisms, it is the only process of respiration. Okay? Glycolysis occurs in the cytoplasm of the cell. During the process, glucose gets partially oxidized. In plants, this glucose is derived from sucrose or from storage carbohydrates. During the course of process in plants, this sucrose is first converted into glucose and fructose by the action of invertase enzyme. After this, these two monosaccharides enter the glycolytic pathway. Glucose and fructose enter the glycolytic pathway. In glycolysis, a chain of 10 reactions occurs. Often reactions occurs under the control of different enzymes. It involves steps which are very, very important. Step 1, phosphorylation of glucose occurs under the action of an enzyme hexokinase and magnesium 2 plus that gives rise to glucose 6-phosphate by the utilization of ATP. In step 2, isomerization of this phosphorylated glucose 6-phosphate takes place to form fructose 6-phosphate with the help of an enzyme phosphohexose isomerase. In the next step, fructose 6-phosphate is again phosphorylated by ATP in order to form fructose 1,6-biphosphate. Fructose 1,6-biphosphate in the presence of the enzyme phosphofructokinase and magnesium 2+. The steps of phosphorylation of glucose to fructose 1,6-biphosphate activates the sugar, thus preventing it from getting out of the cell. Let's move on to next step. Step 4. Splitting of fructose 1,6-biphosphate takes place into 2-triose phosphate molecules. That is dihydroxyacetone, diacetone, 3-phosphate and 3-phosphoglyceroldehydrate, PGAL. This reaction is catalyzed by an enzyme adylose. Step 5. Each of the molecule of PGAL removes 2 redox equivalent in the form of hydrogen atom and transfer them to a molecule of NADP and accepts inorganic phosphates from phosphoric acid. This reaction in turn leads to the conversion of PGAL to 1,3-biphosphoglycerate. Step 6 is the 1,3-biphosphoglycerate is converted to 3-phosphoglycerate with the formation of ATP. This reaction is catalyzed by an enzyme phosphoglyceride kinase. It is also known as energy yielding process. The formation of ATP directly from metabolite constitutes substrate level phosphorylation. Reversible reaction. This is a reversible reaction. Step 7. In this step, 3-phosphoglycerate is subsequently isomerized to form 2-phosphoglycerate. Catalyzed by enzyme phosphoglyceromutase. In step 8, in the presence of enzyme enolase and magnesium Mg2 with the loss of the water molecule. Phosphoglyceride is converted to phosphophenol pyruvate. In step 9, high energy phosphate group of phosphophenol pyruvate is transferred to a molecule of ADP by the action of enzyme pyruvate kinase in the presence of magnesium 2+ and potassium. This in turn produces 2 molecules of pyruvic acid and a molecule of ATP by substrate level phosphorylation. The pyruvic acid thus produced is the key product of glycolysis. In the following slide, the overall reaction of glycolysis can be depicted as glucose plus 2Pi plus 2ADP plus 2NAD, 2 pyruvate plus 2 ATP plus 2 NADH plus 2H. 2 molecules of NADH on oxidation produces 6 molecules of ATP. Therefore, a net gain of 8 ATP molecules occurs during glycolysis. This is what they'll ask in MCQ, not the whole step. Okay. The fate of glycolysis depends upon the availability of oxygen in the cell. In the presence of oxygen, pyruvic acid will enter the mitochondrion and undergo complete oxidation, undergo complete oxidation of glucose to carbon dioxide and water in anaerobic reaction. On the other hand, in the absence of oxygen, the pyruvic acid will undergo anaerobic respiration. Various microorganisms, bacteria, animals and plants are known to catalyze pyruvic acid into various organic compounds depending upon the specific enzymes they possess. Whatever enzyme they have, they will produce different compound. Okay. Some of these types are: number one, during alcoholic fermentation in fungi. That is yeast, and some higher plants, the incomplete oxidation of glucose is achieved under anaerobic condition by a series of reactions. So now we are going to see that reactions in which pyruvic acid is converted to carbon dioxide and ethanol. Ethanol is alcohol. It is done under two steps. Pyruvic acid is first deoxylated to acetaldehyde formed in the presence of enzyme pyruvic acid decarboxylase, that is CH3CCOOH to CH3CHO plus carbon dioxide, that is acetaldehyde. This acetaldehyde is further reduced to ethyl alcohol or ethanol in the presence of enzyme alcohol dehydrogenase. Yes. So that's ethanol. Ethanol is our real alcohol. So body itself produces alcohol from pyruvic acid. During lactic acid formation, during lactic acid fermentation occurs in the muscle. Organisms like some bacteria produce lactic acid as an end product from pyruvic acid. During the reduction, the pyruvic acid produced in glycolysis is reduced by NADH2 to form lactic acid. Carbon dioxide is not produced and NADH2 is oxidized to NAD+. This reaction is catalyzed by a lactic acid dehydrogenase FMN protein and zinc 2 plus ion. Likewise, in case of animal cell, also during exercise, when there is inadequate amount of oxygen for cellular respiration, pyruvic acid is reduced to lactic acid in the presence of enzyme lactate dehydrogenase. Thus, in both the process, oxidation of reducing NADH plus H plus agent takes place. In both alcoholic and lactic acid fermentation, the energy released is very less. Okay, not more than 7% of energy is released from glucose, and not at all of it is trapped as high energy bond of ATP. Also, the fermentation process are proved to be hazardous in nature because either acid or alcohol is produced on oxidation. Apart from this, yeast may also poison themselves to death if the concentration of alcohol reaches about 13%. Aerobic respiration is the next step that leads to complete oxidation of organic substance. It occurs in the presence of oxygen. The oxygen acts as a final acceptor of electron and proton and are removed from the substrate. The final product of glycolysis, that is the pyruvic acid, is transported into from cytoplasm, mitochondria, and thus the second phase of respiration is initiated. Second phase of respiration is initiated. The process of aerobic respiration involves two crucial events. The complete oxidation of pyruvate occurs by the stepwise removal of all the hydrogen atoms, thereby leaving three molecules of carbon dioxide. This occurs in the matrix of mitochondria. The electrons removed as part of hydrogen atom are then passed on to molecular oxygen with the simultaneous synthesis of ATP. This, on the contrary, takes place on the inner membrane of the mitochondria. In the next slide, in mitochondria, pyruvic acid forming a key component. It's a key component. Acetyl-CoA by the action of pyruvic acid dehydrogenase through a series of reaction. Pyruvic acid plus CoA plus NAD plus magnesium 2 plus, which will give acetyl-CoA plus pyruvate dehydrogenase, carbon dioxide, NADH and hydrogen ion. This acetyl-CoA acts as a connecting link between glycolysis and citric acid cycle. During this process, two molecules of NADH are produced by breaking down of two molecules of pyruvic acid. The acetyl-CoA then enters a cyclic pathway, Krebs cycle in the mitochondrial matrix. Various coenzymes including NAD and CoA also participate in the reaction catalyzed by pyruvic acid dehydrogenase. Okay, let's look at the Krebs cycle steps. Steps of Krebs cycle. First, condensation. Remember the word condensation. The Krebs cycle starts with condensation of acetyl group with oxaloacetic acid and water to yield citric acid. Oxaloacetic acid and water to yield citric acid. 6C compound. This is the first stable product of the cycle. This step is catalyzed by an enzyme citrate synthetase. CoA is liberated during this reaction. Citric acid then undergoes reorganization in two steps in order to form, in the presence of an enzyme, cis-acetinase intermediate. Next, look at this, oxidative decarboxylation. Isocitrate is followed by two successive steps of oxidative decarboxylation that leads to the formation of alpha-ketogluteric acid and then succinyl-CoA catalyzed by a ketogluteric acid. The succinyl-CoA then splits into a 4C compound, succinic acid and CoA, with the addition of water. During this conversion, a molecule of GDP is synthesized by an enzyme succinyl-CoA synthetase. When coenzyme A transfers its high energy to a phosphate group, add to the GDP to make GTP molecule. In the remaining steps of the Krebs cycle, succinyl-CoA is oxidized to oxalic acid, a 4C compound, following the formation of fumaric acid and malic acid catalyzed by enzymes succinate, dehydrogenase and fumarase respectively. Now look at the next slide. During this cycle of reaction, 3 molecules of NAD are reduced to NADH, H plus, and 1 molecule of FAD is reduced to FADH2, and also 1 molecule of ATP is reduced directly from GTP for continuous oxidation of acetyl-CoA. Continued replenishment of oxalic acid is necessary. Oxalic acid is necessary. In addition to this, regeneration of NAD and FAD from NADH and FADH2 are required. So the summary reaction for this phase of respiration is pyruvic acid plus 4 NAD plus FAD plus 2H2O plus ADP plus P1 mitochondrial matrix, which gives 3 carbon dioxide plus 4 NADH, 4 hydrogen ion, FADH2 plus ATP. Till now, glucose has been broken down to release carbon dioxide and 8 molecules of NADH, hydrogen ion, 2 FADH are synthesized and just 2 molecules of ATP are produced. Now let us look at the citric acid cycle. The important steps here is, this is the major pathway for formation of ATP molecules. A major pathway for the formation of ATP molecules. Many intermediate compounds of this cycle are used in the synthesis of other biomolecules. In the next cycle, electron transport system is the metabolic pathway through which the electron passes from one carrier to another. It occurs in the inner mitochondrial membrane. The electron transport system consists of 4 enzyme complexes. Which are arranged in a series of inner membrane of the mitochondria. Electrons from NADH produced in citric acid cycle are oxidized by NADH dehydrogenase. Complex 1 and are transferred to ubiquinone located within the inner membrane of the mitochondria. The next step is reducing equivalent are also received by ubiquinone via FADH2 generated during the oxidation of succinate in the citric acid cycle. Next, during the course of transfer, when electron passes from one carrier molecule to another in the electron transport chain, they get coupled to ATP synthesis for the production of ATP from ADP and inorganic phosphate. The number of ATP molecule synthesis depends on the nature of electron donors. One molecule of NADH on oxidation provides 3 molecules of ATP, while one FADH2 produces 2 molecules of ATP because its redox potential is higher than NADH, redox potential, and thus enters the ETS, that is electron transport system, after bypassing the first site of phosphorylation. the electrons are finally accepted by oxygen having the highest redox potential in the series, which along with hydrogen ions form water. Aerobic process of respiration takes place only in the presence of oxygen. The role of O2 is limited at the terminal stage of the process. Yet, the presence of oxygen is vital because it drives the whole process by removing hydrogen from the system. Now look at what is oxidative phosphorylation. Oxidative phosphorylation in mitochondria is catalyzed by a multi-component enzymatic complex called ATP synthesis or coupling factor. The ATP synthesis complex consists of two major components. First is the F1 particle. The headpiece is peripheral membrane protein complex and contains the site for ATP synthesis from ADP and inorganic phosphate Now, F0 particle, it is an integral membrane protein complex that forms the channel through which proton cross the inner membrane. The passage of proton through the channel is coupled to the catalytic site of the F1. The F1 component for production of ATP. Catalytic site of the F1 component for production of ATP. For every 2 hydrogen ion that passes through F0 from the intermembrane space to the matrix, 1 ATP molecule is produced. Okay. Coming to the next slide, the complete aerobic breakdown of 1 molecule of glucose results in the release of 2 molecules of NADH from glycolysis and 8 molecules of NADH and 2 molecules of FADH within the mitochondrial matrix. The calculation of net gain of ATP for every glucose molecule oxidized can be made only on certain assumption. We just have to assume how much ATP is doing. All pathway function in an orderly sequential manner with one substance forming the next with glycolysis, tricyclic acidic cycle and electron transport system pathway, following one after the other. NADH synthesized during glycolysis, it transferred into the mitochondria for oxidative phosphorylation. None of the intermediates in the pathway are utilized to synthesis any other compound. Only glucose is being respired. No other alternative substance are entering into the pathway of any of the intermediate stages. But this kind of assumption are not exactly valid in a living system due to following reasons. What are the reasons? Let us see. This kind of assumption are not exactly valid in the living system. The reasons are all pathways do not take place one after another. Instead, they work simultaneously. Substances tend to enter and withdraw from the pathway whenever required. Utilization of ATP takes place at the time of need. Next, rate of enzyme action are controlled by multiple means. In spite of all these reactions, it is necessary to do this exercise in order to appreciate the beauty and efficiency of the living system in exactly and storing of the energy. Due to all criteria mentioned, it is predicted that there can be net gain of 36 ATP molecules from one molecule of glucose during aerobic respiration as given below. 36 ATP molecule from one molecule of glucose. Glycolysis 2 ATP, which is 2 ATP directly. 2 molecules of NADH, 6 ATP so far. Now, pyruvic acid, you get 2 molecules of NADH, that is 6 ATP. 2 acetyl-CoA citric acid cycle, 6 molecules of NADH, that is 18 ATP. That is equal to 2 molecules of FADH, 4 ATP, 2 molecules of GTP and 2 ATP. So total 38 ATP, 38 ATP. In eukaryotic cells, 2 molecules of ATP are spent for transporting 2 molecules of NADH formed during glycolysis to the mitochondria. Hence, the net gain is 36 molecules of ATP. So, 2 molecules is spent. So, 38 is produced, but eventually we get 36. Fermentation, aerobic respiration. We need to understand the difference between them. What is fermentation and the difference in the aerobic respiration? In fermentation, it accounts for only a partial breakdown of the glucose. Remember the word partial. In aerobic respiration, it accounts for complete degradation of glucose to carbon dioxide and water. What is the second difference? In fermentation, there is net gain of only 2 molecules of ATP for each molecules of glucose degraded to pyruvic acid. Here, in aerobic respiration, in this, many more molecules of ATPs are generated. That is, up to 38 ATPs are generated in aerobic respiration. Next, what is the difference? In fermentation, electron transport is absent. Here in aerobic respiration, it involves electron transport system. It involves the electron transport system. The term amphibolic is used to denote a biochemical pathway involving both catabolism. Example, the citric acid cycle acts as a central metabolic pathway, completes the oxidative degradation of monosaccharides, fats and amino acid. Usually, glucose is the primary substrate for respiration for most of the organism. All carbohydrates are first converted into glucose before they are used for respiration. Different substrates like fat and protein can also be respired, but they do not enter the respiratory pathway at the very first step. If fats have to enter the pathway, they first have to be converted into fatty acid and glycerol. Fat has to be converted, then it has to be converted into fatty acid and glycerol. Fatty acids are then degraded into acetyl-CoA, which enters the citric acid cycle. Glycerol is first converted to 3-phosphoglycerol glyceraldehyde PGAL before entering the pathway. Likewise, the protein are degraded by proteinase into individual amino acid, which after deamination enters the citric acid cycle either as pyruvate or acetyl-CoA. Thus, it is clear from the discussion that respiration involves breakdown of organic compounds. It has been considered as catabolic process, and the respiratory pathway is called catabolic pathway. Respiratory pathway is called catabolic pathway. Remember catabolic pathway. Cat is like breaking down. But at the same time, certain intermediates of respiratory pathway are also withdrawn in order to synthesize other compounds. Example, fatty acid would be broken down to acetyl-CoA, but when organism needs to synthesize fatty acid, acetyl-CoA would be withdrawn from the respiratory pathway. Hence, respiratory pathway comes into picture both during breakdown and synthesis. Breaking down process within the living organisms. Breaking down process within the living organism is catabolism, and synthesis is anabolism. Thus, it would be better considered the respiratory pathway as an amphibolic pathway rather than catabolic one. Amphibolic pathway rather than catabolic one. And that is the difference between fermentation and aerobic respiration. Respiratory quotient is the ratio of volume of carbon dioxide evolved to the volume of oxygen consumed in respiration. It is also known as respiratory ratio. That is, RQ is volume of CO2 evolved divided by volume of oxygen consumed. The respiratory quotient tends to vary with the respiratory substrate used during respiration. This can be understood from number 1, respiratory quotient of carbohydrate, number 2, respiratory quotient of fats, and number 3, respiratory quotient of protein. Let us look at one by one. The respiratory quotient of carbohydrates, when carbohydrates are used as substrate and are completely oxidized, the RQ is always number 1. Respiratory quotient is always number 1. The reason is the amount of carbon dioxide evolved is always equal to amount of oxygen consumed. If you look at this equation, C6H12O6 plus 6O2 gives 6CO2 plus 6H2O plus energy. So, RQ is equal to 6CO2 divided by 6O2, that is equal to 1. At the same time, if you look at the respiratory quotient of the fat, if the respiratory substrate is fat, then RQ of the respiring cells will be less than 1 because the volume of CO2 evolved is quite less in comparison to volume of O2 being consumed. See the equation now. For example, fatty acid, tripalmitin, if used as a substrate, that is 2 within brackets C51 H98 O6, that is a fat, which will require 145 O2 molecules, and that will release 102 carbon dioxide, 98 molecules of water. So RQ is equal to 102 CO2, 102 molecules of CO2 divided by 145 molecules of oxygen. So that is equal to 0.7. So for fat it is 0.7. Similarly, if you look at the protein, the respiratory substrate in protein, the RQ is variable. It varies from 0.5 to 0.9 depending upon the oxygen content of the substrate.